All right, so welcome to another deep dive. This time, I think we have to talk about Dr. Rosario's BIO259 lecture
Oh my God,
on
yes.
action potentials.
The one from yesterday.
Yes.
Yeah.
I mean, how does he make ions moving across membranes sound so fascinating?
I know. Honestly, I was kind of dreading that lecture, but
Yeah.
he's just so engaging.
Yeah, I know.
Like, I don't know how
also on the edge of my seat.
So interesting.
Yeah.
Yeah.
Like I was actually excited to learn more about it.
I know, me too. I was texting my roommate, you will not believe what I just learned about neurons.
And especially the way he connected the chemistry with the electrical stuff.
Yeah, thanks. I mean, I personally think that all this stuff is actually really fascinating. So I'm always happy when I'm able to explain some anatomy and physiology to you all.
Yeah. And will you talk about the domino analogy?
Oh, yeah.
I thought that was the perfect way to explain it.
I know. I have never thought about it like that before. It just makes so much sense.
Sure. I mean, so when I think of action potential propagation, I imagine that the entire axon is just like a bunch of dominoes that are standing right next to each other in a line. And in this analogy, we can imagine that each domino that falls over represents one single action potential.
Right.
And when one falls, it triggers the next one?
Yes.
Yeah, so that's what we actually call action potential propagation. It's not just one domino falling, it's that when one falls, it causes the next one to fall, and so on and so on.
That's basically what's happening with action potentials. Each section of the axon triggers the next one down the line.
Right, like a chain reaction. And you said it all starts with that resting potential of known as 70 millivolts, right? It's kind of crazy to think that our neurons are constantly maintaining that electrical imbalance even when we're not doing anything.
Exactly. It's all thanks to the sodium-potassium pump working tirelessly in the background, pumping sodium ions out and potassium ions in to establish that concentration gradient.
Oh, yeah, the sodium-potassium pump, it's like
I was going to say.
the unsung hero of the nervous system.
Yeah.
Yeah. It's doing a lot of heavy lifting, that's for sure.
So much work.
But, Dr. Rosario, I have a question. Why is the resting potential negative in the first place? Why not just zero?
Well, remember that charges are going to separate on their own with facilitated diffusion through a semi-permeable membrane. Positive K ions are going to leak out of the cell, while large negatively charged proteins, well, they're stuck inside the cell. This naturally leads to a negative membrane potential when the cell is at rest.
Okay, that makes sense.
It-
But what about those incoming signals that a neuron receives? Like when you stub your toe, for example. Do those signals always trigger an action potential?
Well, I mean, if you stub your toe and you feel it, then yeah. The reason your toe hurts is because the sensors triggered action potential propagations all the way down to the central nervous system. But remember, this only happens if that first domino falls. This only happens if you meet threshold potential.
Oh, yeah.
Negative 55 millivolts.
Right.
Exactly. If those incoming signals don't depolarize the membrane enough to reach negative 55 millivolts, the neuron won't fire.
So it's like a failed initiation.
Exactly. Not every signal is strong enough to trigger an action potential. The neuron has a built-in filter to prevent it from responding to every little blip. Reaching that threshold, it's like flipping a switch. It triggers the opening of those voltage-gated sodium channels.
And then the sodium ions?
They come rushing into the neuron, driven by both the electrical gradient and the concentration gradient.
Like a tidal wave of positive charge.
Yeah,
Exactly. And all those positive charges flowing in cause that rapid depolarization, shooting the membrane potential way up to plus 40 millivolts.
So it goes from negative 70 to plus 40. That's a huge jump.
And remember, this is the major feature of an action potential. It's that quick, fleeting moment where the membrane potential goes from negative to positive. That signals that the cell is actually active.
But it doesn't just stay positive forever, right? What brings it back down?
Well, that's where potassium comes in. Remember those voltage-gated potassium channels?
Oh, yeah, they were like the bouncers at the party, right?
Exactly. They're a bit slower on the uptake than the sodium channels, but once that membrane potential hits plus 40 millivolts, they start swinging open.
And then what happens?
Well, potassium ions are positively charged, so when those channels open up, they rush out of the neuron down their concentration gradient.
And that outflow of positive charge is what brings the membrane potential back down.
Precisely. That's what drives repolarization, bringing the membrane potential back towards that resting state of anically 70 millivolts.
This is a roller coaster ride for the membrane potential.
Up with sodium, down with potassium.
And you said there's a little dip below the resting potential, right? What was that called?
You're referring to hyperpolarization, or making the membrane potential more negative than when we started. This is part of the action potential that is sometimes referred to as the refractory period.
Refractory period, that sounds
It is. It's
yeah.
a brief period of time where the neuron is basically unresponsive to further stimulation.
So can't fire another action potential right away.
Exactly. That refractory period is crucial because it prevents the action potential from traveling backwards. It ensures that the signal propagates down the axon in one direction only.
Oh, that makes sense. Like a one-way street for the signal.
So like sodium in, depolarization, potassium out, repolarization, little dip for the refractory period, and then we're back to where we started.
Almost. We can't forget about the sodium-potassium pump. It's been working diligently in the background this whole time, restoring those ion gradients back to their resting state.
Oh, right. It's like the cleanup crew, making sure everything is tidy for the next party.
The unsung hero strikes again.
Absolutely. Without the sodium-potassium pump, the neuron wouldn't be able to fire repeated action potentials.
And if we go back to our domino analogy for a second, the sodium-potassium pumps help with resetting the line of dominoes, right? We're spending energy to stand each one back up.
So that's the whole action potential process. But how do we go from an electrical signal in a neuron to an actual muscle contraction?
Yeah, that's what I was wondering too. It's not like the neuron reaches out and physically tugs on the muscle, right?
Right. There's a bit more to it than that.
I was thinking that maybe it has something to do with that neuromuscular junction you mentioned.
Exactly. That's the meeting point between a motor neuron and a muscle fiber.
So it's like where the electrical signal gets translated into a chemical signal.
Precisely. When the action potential reaches the axon terminal of the motor neuron, it triggers the release of a neurotransmitter called acetylcholine.
And remember, acetylcholine is that neurotransmitter. It is abbreviated as ACH or sometimes just AC.
AC is a key player in muscle contraction. It diffuses across the synaptic cleft, the tiny gap between the neuron and the muscle fiber.
And what does it do when it reaches the muscle fiber?
Well, the muscle fiber has these specialized receptors for AC called acetylcholine receptors.
So it's like a lock and key mechanism.
Exactly. When AC binds to those receptors, it causes a change in the muscle cell membrane.
And that changes.
It opens up channels that allow sodium ions to flow into the muscle cell.
And just keep in mind, allowing ions, which are charged in their nature, allowing those to pass through the cell plasma membrane usually indicates a change in membrane potential, which is, by definition, an electric signal. Sodium ions are positive, so if they flow into the cell, they're going to depolarize the membrane.
Sodium ions again. So we're back to electrical signal.
You got it. That influx of sodium ions depolarizes the muscle cell membrane, triggering an electrical signal that spreads across the entire muscle fiber.
So the muscle cell basically generates its own little action potential.
Exactly. And this electrical signal in the muscle fiber is what ultimately leads to the muscle contraction.
So, I mean, that's one of the major points I've been trying to make in the last couple of lectures. You need to understand how neurons work to fully understand how we have so much control of our skeletal muscles. But here's the big question. Do you remember how an electric signal actually causes a muscle to contract?
Whoa! Remember the sarcoplasmic reticulum, that specialized network of membranes inside the muscle fiber? It's basically a storage facility for calcium ions.
Oh, right. Calcium is important for muscle contraction.
Absolutely crucial. But how does the electrical signal tell the sarcoplasmic reticulum to release its calcium?
Yeah, that's what I was wondering.
That's where the T-tubules come in. They're these invaginations of the muscle cell membrane that carry the electrical signal deep into the muscle fiber.
So the T-tubules are like a delivery system, bringing the signal right to the sarcoplasmic reticulum.
Exactly. And when the sarcoplasmic reticulum senses that electrical signal, it releases its stores of calcium ions into the muscle fiber.
And then the calcium does its magic and makes the muscle contract.
Precisely. Calcium is like the key that unlocks the contractile machinery of the muscle fiber.
Unlocks machinery. What does that even mean?
Well, it means that calcium binds to specific proteins on the actin and myosin filaments, which are the proteins that make up the muscle fiber.
Yep, and specifically we're talking about calcium binding to troponin, which will inevitably pull off tropomyosin. And that is what allows the myofilaments to interact with each other.
Actin and myosin, those are the ones that slide past each other.
Yeah. Exactly. And when calcium binds to those proteins, it allows them to interact and slide past each other, which is what causes the muscle to shorten.
Oh, right. The cross-bridge cycle. That's how the muscle generates force.
It's all starting to come together now. Okay, my brain is officially fried. Can we, try to summarize everything?
Yeah, please. I need a recap before I forget everything.
So starting with the neuron, right?
Yep, that resting potential of negative 70 millivolts. Remember, the sodium-potassium pump is working constantly to maintain that,
Pumping all those ions.
like a tiny battery, always charged up.
And then if a stimulus is strong enough to reach that threshold of negative 55,
Boom. The voltage-gated sodium channels open.
and sodium ions rush in.
Exactly, leading to depolarization.
That rapid upswing in voltage, so cool.
But it doesn't stay depolarized forever. Potassium channels come in to save the day.
Right. Repolarization, bringing the membrane potential back down.
Yeah.
And we get that little dip below resting potential.
Hyperpolarization.
Yes, leading to the refractory period.
Ensuring that the signal can only travel in one direction.
So smart, our nervous system.
And then, our good friend, the sodium-potassium pump, resets everything.
Getting ready for the next action potential. And remember, all of this happens super fast, especially in those myelinated neurons.
Saltatory conduction, like high-speed train.
Zipping between those nodes of Ranvier. But then we get to the neuromuscular junction.
And things change, right? It's no longer just electrical signals.
Right. It becomes a chemical message.
And it binds to receptors on the muscle fiber.
And then what?
Well, that binding opens up channels that let sodium ions into the muscle cell.
So we're back to sodium and electrical signals.
Yep, depolarizing the muscle cell membrane.
Right, because remember that the sodium ions are positively charged.
So the muscle cell is like making its own action potential.
Exactly. And that triggers the release of calcium.
Oh, right. Calcium is the key for muscle contraction.
Yep, remember that this is the last chemical signal that finally activates the process of muscle shortening.
It's a cross-bridge cycle.
Wow, it's amazing how all of this works together.
From the initial stimulus to the actual muscle movement, like, whoa!
I feel like I actually understand it now. Thanks for explaining it all, Dr. Rosario.
Yeah, of course. I'm always happy to talk about action potentials. And remember, if you have any more questions, you can come see me in office hours.
We definitely will.
And for all our fellow BIO259 students out there, good luck on the exam. We'll see you on the next deep dive.